Okay, so next we'll talk about SkipGNN, predicting molecular interaction with a skip graph network. So this is a joint work by Ke-Xin Huang, Danica Xiao, Lucas Glass, and Marianka Zini. And this is a paper published at Scientific Report, and you can access through this link. There are a lot of networks in all kinds of biological systems. You have disease networks, drug interaction network, even environmental network and social network. They're all kind of very important in modeling biological systems. And there are a lot of success in using graph neural networks to model biological networks. For example, this paper by Marianka Zini and her colleague on bioinformatics, they're modeling polypharmacy side effects with graph convolution neural network. In particular, this drug interaction and drug protein interaction and protein interaction networks, and using a graph neural networks to model the interaction and finally predicting the polypharmacy side effects. And in a very broadly speaking, to apply graph neural network, the key idea is to aggregate information from each node's neighbors. For example, node B will be you can aggregate information from A and C using a neural network, and similarly C can be aggregating information from A, B, and E and F, and those aggregation functions are modeled with a neural network. However, there's the fundamental assumption here that applying this type of graph neural network, that is this homophily assumption. That is, a node will be similar to its neighbors. And however, in biomedical networks, this type of direct similarity may not be true. So you may have to look at SkipSimilarity, right? That's just like a two-hop neighbors, right? A and B are similar, but there's no direct connection between A and B. But through another node C, they are connected via this two-hop pass. That's actually very, very common in biological network. For example, here we can have this drug-target interaction network, right? Drug A and B. They're very similar, but there's no direct interaction between these two. But through a common target one, and they're connected via this two-hop pass. And so A and B in this case are indeed similar, but standard GNN would not be able to model this type of molecular networks directly because a lot of those edges are not present. So you have to rely on this more long-range pass, and it's much harder to model. So SkipGNN, the key idea is quite simple. That is, since we know that biological network often have this SkipSimilarity, that is, two-hop neighbor are kind of important, maybe we can construct this skip graph. That is, construct another graph, this adjacency matrix for the skip graph AS, that's essentially model all these two-hop neighbors. Once you have the skip graph, you can, again, applying graph neural network on this skip graph, and then to learn the embedding of the node edges and so on. So that's the kind of the key idea. But in this paper, they also have a more integrated version that fuse this both skip similarity and also direct similarity in the same graph neural network structure, so that you can model both of this skip similarity and direct connections together. And finally, after this fusion process, the embedding that captures the best combination of both direct and skip similarity can be concatenated and feed into the final decoder to make prediction. For example, predicting whether a specific link should be present. Okay, so now let's look at some of the experiment performance of SkipGNN compared to many different baselines on 4 different tasks: on drug-target interaction, on drug-drug interaction, on protein-protein interaction, and on gene-drug interaction. So overall, SkipGNN achieved best average rank performance comparing to all these different baselines across these 4 tasks, and that shows the robust performance for SkipGNN. And SkipGNN also performed very well when you have a lot of missing data, right? This figure shows as you increase the percentage of missing, right, from 30% to 90%, the gap, the benefit of using SkipGNN become more and more when you have more missings. And finally, we used SkipGNN to try to identify some novel drug-drug interactions that are, and then we look for some more recent publications, and then this is top 10 most confident interaction that the algorithm identified. Out of this 10 top ranked interactions, we're able to find 6 pairs that have literature evidence support that shows the SkipGNN are indeed very useful for identifying novel drug-drug interaction. So to summarize, we talked about a paper for predicting molecular interactions with skip graph networks. If you're interested, you can check out the paper and also the code on GitHub. Thank you very much.